in something that's more dependable. So I apologize for that. So we're just going to get right back into it. What we were talking about is forming polysaccharides, right? Taking the monomeric carbohydrate subunits, or they could be saccharide subunits if they're substituents of carbohydrates, right? So substituents meaning they modify it from the basic CH2O formula. We start putting amines, carboxylic acids, other kinds of substitutions. And we can start to link them together. And I showed you it's basically a dehydration reaction, right? We just lose water during the process, and what we form is what we call a glycosidic bond or glycosidic linkage. Those can be either O-link, if it's an oxygen that connects to carbohydrates, or they can be N-link, if it's a nitrogen that connects to the two carbohydrates. Okay? We look at two kinds, but remember, when you're building macromolecules, you need energy to drive two molecules together to become one, because there's an entropic penalty for that, right? And for the formation of polysaccharides, what happens is first you phosphorylate it, they use ATP, there's an enzyme. Every time you see a reaction, remember there's an enzyme performing it. Now this is the important point. The enzyme only changes the rate of the reaction, not the thermodynamic properties of it. It doesn't change the favorability of the reaction, right? It only changes the rate at which it happens. So every little reaction that you see in biochemistry that I draw out, remember, it's that chapter 8 talking about enzymes. They are catalyzing all these things, increasing the rate, but not changing the thermodynamics. They're not changing the stability of the reaction. Okay? So first what happens, we use free energy in the form of ATP, phosphorylate. Then you need to phosphorylate it more to make it more activated. This has some free energy. It's not a great amount. So what happens, you bring in UTP, uridine triphosphate, right? And then what happens, the UMP gets added to that phosphate. So you can see we get UMP added, so we have what we call UDP ribosylated this, because uridine has ribose. So we call that UDP ribosylation. And by doing this, now we have free energy to drive the reaction, right? From this, we get now a negative delta G for driving the reaction, for driving glycosidic bond formation. And that's what's going to allow us to build up these macromolecules. Okay? We talked about there being different kinds of polymers that you can make. You can make linear polymers, or you can make branch polymers when we're talking about these six-membered rings. They can be all of one type of carbohydrate, as shown here, or they can be heteropolysaccharides, meaning they're a mixture of different carbohydrates. This is a common way of showing this. So carbohydrate chemists often use color coding in six-membered rings for six-membered carbons, and each of these colors actually represents a different carbohydrate. You'd have to have a key to know what red stands for, blue stands for, but that's a common way that they'll present it. Okay, so it can form linear or branched polysaccharides. It can be a mixture of different ones or the same one. Okay? And we talked about how these connections work. So when we get polysaccharides, we can have linear segments. Remember that there's a non-reducing end. Who remembers what the non-reducing end? Why is it called the non-reducing end? Remember from the directionality? Crickets. Okay, why do we call the other end the reducing end?
You can continue to add.
Yeah, it has an anomeric carbon that can still open up. It still has a hemiacetal, basically. Where you have the hemiacetal, it hasn't had anything attached to that anomeric carbon, and because of that, it can ring open. And because it can ring open, it can undergo a reaction that reduces copper. So we call it the reducing end. The other end does not have an anomeric carbon that's free. The anomeric carbon is locked, right, in a glycosidic bond. And once it gets locked, this is an important point. Once it forms that glycosidic bond, it can no longer bend over. It's stuck in the cyclic form and it's locked there. So we call that in the non-reducing end. Okay, so you can have these long linear segments, okay? And then you get branch points, and these are usually through 1,4 connections. They can be through 1,1 connections as well. This is showing amylose, it's a 1,4 connection. And then you get branch points typically through that 6 position, which branches off, okay? So here's our branch points. This is an alpha 1,6, and this is the structure or the polymeric form of what makes up amylose, which is found in starch, glycogen, those kinds of structures. Now, in reality, we don't have the Hayward projection. This doesn't really tell you the actual 3D structure, right? It actually, these 6-membered rings are in chair conformations. So when you see this, you got to think, oh, these are actually forming chair conformations, and these lead to unique 3D structures. Okay. I always draw them out in the Hayward projection because it makes it nice and flat, easy to draw on a piece of paper. It's just useful for that. Okay. So we talked about a little bit more about that chair conformation and what it does, okay? Depending on the anomeric carbon, this is why we spent some time talking about that anomeric carbon and how it can flip between different anomeric forms. Okay? Whether you have beta anomeric form forming the glycosidic bond or an alpha form, it totally changes the direction of the carbohydrate polymer. Okay? So you can see when we have a beta linked, so this is glucose, this is a beta glucose, and now we're linking a beta glucose, the anomer form, with a 4 position here. And what happens is you get 2 flat chairs that hydrogen bond in this fashion, so you get these flat sheets that come from that. Now, if you just flip that bond, right, we're changing the anomeric form, and now we're connecting an alpha anomeric form of glucose, okay, in a 1,4 alpha connection. Now what happens, it bends this, it tilts it. You can see it's no longer flat like this. And because of that, this leads to things like helicity in the polymeric form. It causes it to bend, and then now we get a new 3D structure. Remember that the difference is, it's just like protein folding. We have phi and psi bonds, but now the phi and psi bonds are between these carbon-oxygen bonds that form the glycosidic bond. Okay? And this changing of the anomer form changes the directionality of those building blocks. The branch points have an extra degree of rotation, so they have an omega bond, and again, those things rotate. And then when you look in 3D, what's happening is we have this folding effect, just like with protein. Okay. Now, one of the things that stabilizes certain structures are hydrogen bonding networks. I didn't talk about that last time. So you can see these hydrogen bonding networks that can help to stabilize certain structures. Okay. So we talked about that last time, and then that led into, let's look at some examples of polysaccharides, so long-chain carbohydrates, what their function is, and where does that function always come from? From the overall structure, 3D structure. So we're going to start with a macromolecule, a polysaccharide, that's very important to all of us. This is the structure of glycogen. It's also the structure of starch. So the storage of sugar in potatoes is very similar to the storage of sugar in our muscle. Glycogen and starch actually have a very similar kind of polysaccharide that make them up. Here's a schematic of the structure of glycogen. It forms these nanoparticles that basically look like whirlwinds. Okay? So you can see there's a central point that it starts from here, and then you see a branch, and then off of this linear polymer you see a branch, and another branch, and another branch. So you get this kind of pinwheel or branching effect, and we call this a whirlwind structure. Now, all the branch points are alpha 1,6 linkages, so that's our branch points. This is our branch. And all the linear parts are through alpha 1,4 linkages. So here, I'm showing the linear parts. We have an alpha, 1,4. Remember, it's on the opposite side. That distinguishes alpha, 1,4. And so we have these long branch parts that are alpha 1,4. Then we have branch points that are alpha 1,6. And what happens is you get this helical structure. So imagine these forming helices. And they're branching off from helices. So you're getting this whirlwind structure. And this structure is useful for storage of energy. Glycogen is what stores sugars, carbohydrates in our muscle. Okay? And it stores it up in this structure. Now, how does this structure lead to the function? Remember, function is energy storage. Function equals energy storage. And here's actually looking in the muscle. Here's a glycogen granule. See these red dots here? Those are nanoparticle structures. There are actually these little dots in there. Those are nanoparticles of glycogen in the muscle. Okay? So why does this structure lead to a function of storing energy? Well, what do we want to do? If we have energy, I talked about this a little bit last time, we want it stored long term so it doesn't get used up. But when we want to take it out and use it, we want to be able to access it readily, right? You want to be able to go down to your bank and get out your money, these during normal business hours whenever you want. But you want it to be safe there so that you don't lose it, right? That it's not in your couch at home and somebody steals your couch and you lose everything. So what's happening here? This structure is very useful because look at all the ends of linear polymers of glucose. So each of these is glucose. You see there is lots of exposed glucose. So there's like 1, 2, 3, 4, 5, 6. You've got probably 30, 40, 50 exposed chains. And each one of those chains can be degraded readily. How glycogen is degraded is one glucose from a linear chain at a time. If you only had one long chain, what would that mean? You could only degrade from one chain. But by branching it out and having a bunch of chains kind of off of this foosball kind of looking structure, you have lots of exposed chains of glucose. So enzymes can come in and they can start degrading at all those different chains. And you can rapidly release them. Okay? So it starts to break down these alpha 1,4 linkages. We have lots of exposed glucose so that we can release glucose rapidly. We'll talk about the signaling that's involved to allow that release, but that's why this is important. So it forms these helical structures too. Now, the helical structure gives it access as well. So these chains aren't just stacked on top of one another, but they're helices, and that keeps them separated so they're exposed inaccessible. Starch is the same. Starch is made up of alpha 1,4 glucose, alpha 1,6 linkages. So the same way we store energy in our muscle cells, a potato basically is storing energy in the same form, in the same kind of structural arrangement. Now let's look at an alternate function. So that was energy storage. We need to be able to access the energy in the form of glucose. Okay? But we need to store it up so it's not just really floating around. Okay. What about sugars that are used for structural purposes? So sugars are used for energy, and they're also used to build up unique structures, like the wood that you see that makes up a tree. Okay? Or the blades of grass. They're made up of largely cellulose. Not all of it is cellulose, but that's the main component that makes up the tree trunk, that makes up blades of grass, cellulose. How is cellulose different? This is cool. It's glucose. The only difference is that it's the beta anomeric form that makes up the monomers. So we have a beta 1,4 linkage that connects them. Okay. Now what was the difference when we looked at that subunit? It forms linear structures, right? Not helical, but because of the hydrogen bonding, it forms flat structures. Okay? And because of this, it will form long sheets. So you have alpha 1,4, and what happens is you get these linear sheets, and in cellulose, in plants, they stack sheets of cellulose on top of one another, connected through hydrogen bonding networks, and that leads to structural stability. I'm going to borrow something real quick. Can I borrow your notebook real quick? Okay, sheet of paper. How can we understand? Why do we put sheets? And what do we know about wood? It's very strong, right? Takes a lot to break it. It's like a sheet of, okay, one cellulose is like a sheet of paper. Easy to tear. Now, if you stack sheets of cellulose on top of one another, what happens? You can't tear it. It provides strength. It provides rigidity. Sorry about the one sheet of paper. That's what's happening here. If you make sheets, one sheet's not very strong, really. But if you start to stack them up and they're nice and flat, you can't tear them. That's how cellulose works, and that's how wood and blades of grass, that's where they get that structural stability. It's from cellulose, and here is an actual picture, a cutaway of a green leafy material, and you can see these lines of cellulose. Do you see them? So even these lines are made up of stacks, and then what happens are these stacks stack on top of one another one way, and then you can see there's a layer below it, they stack another way. So they're alternating the way they go. That provides structural stability. Alpha 1,4, or beta 1,4, structural stability because it's flat. Alpha 1,4, helical, and it's accessible and easy to get to for energy storage. So there's where that comes from. Pretty simple, basic structural difference. Just change the anomeric carbon, get totally different structure, totally different function. Okay, now cool thing, basically half of all the carbon on Earth in the biosphere is locked up in cellulose, because there's lots of plants. So about 50% of the carbon in the biosphere is locked up in cellulose. It provides strength and rigidity, such strength that we can build buildings out of wood and all that kind of good stuff. Okay, now there's a similar molecule that's kind of related to cellulose. It's called chitin. You might have heard it called other things. I call it chitin. And basically it's the same thing as cellulose, except it's a glucosamine that is acetylated. So we have this acetate group that's acetylating it. Okay? And so this is actually a 1,4 linkage. So you can see here's glucose, but now it's a glucosamine, and it's been acetylated. Okay? So this would be glutamic, glucosamine, or amidated. I'm sorry, it's amidated, acetylated, amidated. And so what you have here is a similar structure, but now has a slight chemical modification. But it's actually used for structural materials in insects. So chitin is the main component that makes up the exoskeleton of insects. So this is just a slight modification here chemically, changes its properties slightly, but what it's used for in the insect is to make these sheets, just like in cellulose, okay, that make up their hard, crunchy exoskeleton. Again, this is a beta. Did I say alpha before? I apologize. This is a beta. Beta 1,4. I should say 1. Beta 1,4. Okay? Because see here, this is on the same side. See, here's the CH2 up here. Hydroxyl's on the same side. It's beta. If it was on the other side. Okay? So this is almost as abundant as cellulose. There's 10 to the 14th kilograms of chitin produced annually. Because there are a lot of insects, especially in Texas, right? And we have always spraying for all kinds of nasty little things outside the house. It makes up the exoskeleton of invertebrates. It's using the same strategy as what we see in plants. Okay. Sugars are also, or carbohydrates are also found as important conjugates. So not purely just carbohydrates, but now we can conjugate it to other molecules, other macromolecules, like lipids. We can conjugate them to proteins. Okay, we already know that sugars make up nucleic acid. Okay, so when we put a carbohydrate on another molecule, we call it a glycoconjugate. Okay, remember we have a name for carbohydrates. We also call them glycans, right? So then we get the term glycoconjugate. This is a covalent linking of carbohydrates to proteins, lipids, or other molecules. So if you see a carbohydrate on something other than just carbohydrates, we would call that a glycoconjugate. This is important because it makes up structural components of organisms. A lot of the structural components are glycoconjugates. It's a component of RNA and DNA, which we already know, ribose. But it's very important. It's not just a structural thing. So carbohydrates not only serve structural purposes, they also communicate information. Okay? They're used for communication and recognition. And I'll give you an example of this. For instance, the blood types we'll talk about. That gives information to the cell. So let's look at some examples of glycoconjugates. Okay, here's a figure from a different book. Basically, you can attach, and here's a key, and they're showing the different carbohydrates as a symbol, and then they colorize it. So if you want galactose, that's a sphere with yellow. Glucose, that's a sphere that's blue. And then we can see these acylated ones that we looked at. You can see these are different ones. Mannose. This is typically what carbohydrates chemists do because they have to draw out these chains. And then can you imagine just drawing out all these, even abbreviations to show a linkage? It'd be crazy. So they just draw them out like this, and they give you a key. Okay, so the different kinds of major glycoconjugates, we can have them attached to proteins. And these are often attached to proteins that are anchored on the surface of the membrane. Okay? So we often call these proteoglycans because they're proteins that have carbohydrates added to them. We also can call them glycoproteins. Those terms are pretty much interchangeable. Okay? Now, where do they get attached? They typically get attached, so there are three different ones that they can get attached to. Serine, threonine, and asparagine. Okay, those are the three that they can get attached to, and we'll look at how that works. But a lot of time, all the proteins, or a lot of the proteins that are on the surface of your cell are usually glycosylated. Okay? And they get connected through asparagine, serine, and threonine. Okay? Proteoglycans can interact with extracellular proteins. So this is what I mean by information. They present chemical information that allows other biomolecules to interact with them. Okay? Without those carbohydrates there, they might not interact. Okay? So they interact with extracellular proteins, and they actually bind through electrostatic interactions. Proteoglycans compose the extracellular matrix and can mediate interactions and activities. You can also see that you can have these carbohydrates attached to lipids as well that make up the membrane. And this is where you get the blood types, where you get that section. It's through anchoring of carbohydrates through lipids. So let's look at some examples of these. So these are important, they're important structural components of connective tissue. So chondroitin sulfate, keratin sulfate, okay, so we see galactose, glucose, it's been modified, it's carboxylated. We have some that have sulfates on them. Hyaluronic acid, you may be familiar with things like chondroitin, keratin, hyaluronic acid. If you look at supplements for joints, they often contain chondroitin. We used to give our pigs supplement to help with their joints when they're growing really fast. They could get crippled because they're growing so fast their joints can't keep up. So we would often give them, actually, it's a horse supplement that contains chondroitin. Why do we give them chondroitin supplements? Because their connective tissue, the part that gets damaged when they're growing really fast, uses chondroitin to make up the structure that makes up the connective tissue. So this is what's shown here. This is actually connective tissue, and you can see the carbohydrates that make it up. So your cartilage, for instance, the parts of your joints that kind of that protect it, make it soft, they actually are glycoconjugates. Hyaluronic acid, this is kind of something that's interesting. People often use these in beauty products to make their skin look more wrinkleless. It goes into the surface of the skin, helps to make it look puffy and not wrinkly. So because it makes up those kinds of connective tissues, those kinds of the substructure that gives you that. So proteoglycans make up the connective tissue. They make up things like the bacterial cell wall, peptidoglycan, which we've seen. Remember, lysozyme, that enzyme that we examined, it breaks down the peptidoglycan because it cuts the NAG and the NAM, right? The N-acetylglucosamine and the N-acetyluramic acid. These things make up the peptidoglycan layer, shown here. And what lysozyme does, remember, is it cuts a glycosidic bond between those. Okay? And this is just showing you how that bacterial cell wall looks when you see that peptidoglycan layer right here. Here's again a close-up of the peptidoglycan, so we can see these alternating NAG and NAM, right? These are beta-1,4 link. And what do we remember about beta-1,4? What was it used for?
Very strong.
Strong, right? Which makes sense. If you're making up a bacterial cell wall, you don't want it to be degraded readily, right? And so these are beta-1,4 link, and you can kind of see here how they're connected together through peptides, and they're all linked together and they help provide structural rigidity to the cell wall. And actually, we use this as a way to prevent bacterial growth. We actually target the enzyme that makes peptidoglycan. So here's how that layer is actually made. There's an enzyme called transpeptidase, and what it does is it's responsible for connecting chains of these carbohydrates, these NAD/NAM, and it connects them together via an enzyme called transpeptidase. So if we go back one, you see these peptides that connect these peptidoglycan carbohydrate layers. Okay? And now we're stacking these around. There's an enzyme that does this transfer. It's called transpeptidase. Okay? And the cool thing about bacteria versus us is they have peptidoglycan that make up their cells. We don't. Now because of that, there's a biochemical difference. And when there's a biochemical difference, if you want to kill something, you target the difference. Okay? And that's what we do. They have this enzyme called transpeptidase, and what it does is it takes two chains. So here's one chain, okay, and it connects it to another chain. So it gets activated on this enzyme, and then it connects it. So it's attached to the enzyme and connects it to another chain, and that's how you get the peptidoglycan. When you use penicillin or these antibiotics, what you're actually targeting is the enzyme transpeptidase. So over here, we have penicillin. This is what it looks like. It has a lactam ring. So that's a 4-member ring with an amide right in the ring. And what happens is this actually, the enzyme reacts with this ring. Instead of cross-linking, if you throw penicillin in, what happens? This enzyme actually attacks the lactam ring. Ring opens this lactam so that it basically inactivates the enzyme. It forms a covalent intermediate. You see right here, we no longer have that ring structure. It's been opened, right here, that carbonyl is now attached to the enzyme. And it can't go backwards. It's stuck on the enzyme, and that's how our antibiotics, a lot of them work. They target enzymes that will react with them, and these act as kind of suicide or mechanism-based inhibitors. So, little side steps, that's what happens. Now, because you take out transpeptidase, what can the bacteria not do anymore? Yeah, can't build cell walls, so it can't grow and divide. It'll still be there, but the idea is if you inhibit its ability to grow, what can then come in and help clear it out? Your immune system. So the antibiotics stop the growth And the immune system has a chance to come in and wipe them out. Okay. So here's something about the structure of these kinds of glycoconjugates. Remember, this is a glycoconjugate because they're cross-linking a peptide with these chains of carbohydrates. Okay, let's look at glycoproteins. I said they can be linked through threonine and asparagine, right? How does that work? And this is just showing you how that connectivity works. Serine provides a hydroxyl. And hydroxyls, remember, are used to connect polysaccharides together. So it actually serves as a useful point where we can make an O-linked glycoconjugate. So you can see here serine. You could also imagine threonine. Remember, threonine only just has a methyl group off that carbon, right? So either threonine or serine can be used to make O-linked connections with carbohydrates or saccharides. You can also make N-linked. So asparagine, actually, which has that amide group coming off there, you can actually have an N-link through that. So we use that nitrogen to form a link with, in this case, glucosamine, acetylglucosamine. Okay? So this is just showing you that when we connect these, you can get different kinds of polysaccharides coming off. You can get as little as 1 or 2. You can get branch chain polysaccharides coming off. There are all different kinds of variations of what can be added to proteins, but they get added at a point, either asparagine, threonine, or serine. Okay, these can be very important for functional purposes. Actually, some enzymes and proteins, if they don't get glycosylated, they don't take on the right structure, and they don't function properly. Okay? So these can serve important structural purposes because it becomes part of the protein structure. Eukaryotic proteins are typically highly glycosylated. Most of the eukaryotic proteins, at least in humans, they are glycosylated. Okay, so those are glycoproteins. Let's look at glycolipids. How do these look? So here is actually the lipid part. We'll talk about lipids next. It's funny that we talk about lipids before we even actually talked about them, but those are the hydrophobic molecules like fatty acids. And you can see that we can connect them to carbohydrates, shown here, and then we can get off of them these long chain polysaccharides. Okay. Gangliosides, for instance, are membrane lipids of eukaryotic cells. These contain very complex oligosaccharides. Lipopolysaccharides, LPS, endotoxin, which comes from peptidoglycan, is a dominant feature of the outer membrane of gram-negative cells, bacterial cells. This causes endotoxic shock in humans, and so when people are producing drugs and things like that for people, they have to make sure that there's no LPS because these can trigger strong immune responses. So this is used for identification. Bacterial cells have a different glycolipid layer that can often stimulate immune response. Okay, well-known case of glycoconjugates, sugars that make up the blood cells. So these carry important information. Actually, how does your immune system know self from something else? It's from actually the glycosylation of your cells. Your cells, like your blood cells, have different glycosylation patterns than, say, a bacterial cell, okay? Or what a virus has on its surface. So viruses oftentimes are glycosylated. And actually, that's how they evade the immune system sometimes. They change their glycosylation pattern. But with blood cells, it's the same kind of deal. When you have O, B, A, AB type blood, depends on what polysaccharides are on the surface. These are lipopolysaccharides, so they're anchored through a lipid from the membrane. Okay? So your O link here has a galactose, an N-acetylglucosamine, a galactose, and then connected to that we have a fructose. Now, how is this different than B-type? B-type only is different at one position here. You see this galactose position? We have another galactose that branches off from that ring. that's the difference between O and B type. A and B type, A is different because it has a slightly different saccharide coming off of there. It has N-acetylgalactosamine coming off at that point. And then if you have A, B type, you actually have a mixture on your blood cell surface. Okay? So, why is this important? Because if you want to get a blood transfusion, you have to make sure you get the right type of blood. Because your immune system knows which type of blood you started with. And if you put in, so let's say you have O-type blood, and you throw in B-type blood, you go to the doctor and you throw that in, what happens? The body will reject it, because your immune system says, that is not supposed to be there. My blood does not have it. And so if you're an O-type, you cannot receive B, AB, or A type. Okay? It's bad from that standpoint. Now, so if you are an O-type though, you can actually donate your blood to anybody. Because look at all these structures. Do you see they all have the same starting structure that O does? So we call the O a universal donor. You can donate your blood to anybody and they're going to be fine. Because